Hi, and welcome to the September edition of the Forensic Update. Time is of the essence in forensic investigations when determining how much time has elapsed since a death. This could only be determined after 36 hours until now. Recently, researchers at the University of Salzburg found certain proteins and enzymes in muscle tissue don't break down until after 240 hours after death. This could mean a time of death could be determined in a much longer window since the death occurred. The study was conducted in pigs, the closest animal to humans in post-mortem, and the researchers are currently looking at human samples. Other factors, like the age of the victim, will also have to be studied before this technique can be implemented, but the early results show big promise for forensic science. A fingerprint may be able to place a suspect at a crime scene, but not necessarily at the time of the crime, until now. Two chemists at the National Institute of Standards and Technology, or NIST, accidentally discovered a method for dating a fingerprint. The researchers were trying to study trace amounts of drugs on prints when they realized some chemicals move from the peaks to the valleys of the prints over time. Using a technique called time-of-flight secondary ion spectrometry, the two focused on the migration of fatty acids to predict the age of the print. If approved for fieldwork, this could help investigators narrow down the prints to the ones that matter the most. This month marks the 14th anniversary of the 9/11 terrorist attacks on the United States. The world changed, and so did the application of forensic science. Among other security changes, CSI-style techniques were brought to the battlefield to assist military operations. NFSTC has been proudly training our military law enforcement on how to utilize and apply forensic techniques for nearly 5 years. Tony Tambasco has led forensic laboratories for decades, and he says NFSTC has been a key player in transitioning training to the Department of Defense community.
Well, I see the training to the military right now as being phenomenal. Bringing the expertise of the crime laboratory out to the field, being able to train that aspect of it. I didn't expect that. In a crime lab, you think of chemistry, trace, biology, boom, boom, boom. Now you have to think outside. What else is there? What are there other forensics that we didn't have before that you honestly have to be concerned about?
You can learn more about the training, also called site exploitation, on our website. A new partnership between the Huntington Police in West Virginia and the Marshall University Forensic Science Center could reduce or eliminate the agency's untested sexual assault kit backlog. The university's accredited lab will test for DNA on all of the sexual assault kits from the agency. The resulting eligible profiles will then be sent to the West Virginia State Police to be uploaded into CODIS. This partnership has already resulted in a charge of sexual assault from 2004. With a little tape and patience, you could have your very own spectrometer that fits in the palm of your hand. The folks over at Public Lab have created a DIY version of the analysis tool. Once all the pieces are put together, the spectrometer attaches to the camera of your cell phone and works with web-based software to record data. We put a link to the free downloadable instructions on our As Seen on the Forensic Update page at nfstc.org. If you've been following the work of the Organization of Scientific Area Committees, the OSAC just made it easier for you and will be publishing a monthly newsletter. We placed a link on our website to sign up. That'll do for this edition of the Forensic Update. From all of us here at NFSTC, I'm Bill Duffin, and thanks for watching.